Hello and welcome everyone. This is Dr. Siham Al-Araj. I will be giving you the practical laboratory part of the oral mucosa 1. The oral mucosa can be classified upon the primary function served as masticatory lining or specialized mucosa, or upon keratinization level, either keratinized, ortho or para-keratinized, or non-keratinized, or classified upon the location to buccal mucosa, lingual, palatal, labial, or alveolar. The oral mucosa is composed of an epithelium made up of 4 layers, the lamina propria underneath, which could be divided in 2 parts, the submucosa, which separates the mucosa from the bone or the muscle underneath. The oral epithelium is divided into keratinized or a non-keratinized epithelium. The keratinized part has an uppermost layer of stratum corneum, which is a layer we don't see any cells. The second layer is the stratum granulosum, which we see by the arrows contains granules of phospholipids primarily. The next layer is the stratum spinosum, named after their appearance. And the fourth down layer is the stratum basale, which is just one layer of cells. On the other hand, the non-keratinized epithelium is divided into the uppermost superficial layer, which has nuclei, the intermediate layer, the prickle cell layer or the spinosum layer, and also the basal cell layer, which share its characteristics with the keratinized epithelium. As we can see, in the keratinized epithelium, there is one large layer of almost 20 cell thickness that is keratinized or can be para-keratinized, which contains some nuclei. We see the granular layer is the layer next to it, then the prickle cell layer, and one layer of the basal layer. The basal layer contains the stem cell which undergo mitosis and move for differentiation upward in the other layers. On the other hand, the non-keratinized, we have subdivisions: superficial layer, intermediate layer, the prickle and the basal cell layer as the other type. We can see a microscopic picture of a non-keratinized epithelium, the superficial layer, intermediate layer, prickle cell, and the basal layer. You can easily know what type of epithelium if I ask you. In the non-keratinized mucosa, we see an epithelium with reedy ridges. The lamina propria is composed of irregular bundles, collagen bundles or fibers, which are irregular and loosely arranged. There's a layer of submucosa before the bony layer attachment. On the other hand, some parts of the keratinized epithelium are characterized by more profound and numerous reedy ridges of the epithelium, a thickened lamina propria, which is composed of a lamellar structure of collagen bundles, regularly and structurized. Thickened lamina propria is directly attached to the underlying periosteum and bone. This is called the oral mucoperiosteum, referring to its attachment directly mucosa and the periosteum. The keratinized epithelium is composed of a basal cell layer, which are cuboidal cells that are least differentiated and undergo mitosis to progenerate keratinocytes to the upper layers. Next is the prickle cell layer containing tonofilaments and start of granules. Next layer is the granular layer characterized by the keratohaline numerous granules in the cytoplasm, while the uppermost layer is the keratinized, which is composed of dead cells that contain no nuclei and no granules and are continuously shed because of friction and the masticatory forces. On the other hand, the non-keratinized epithelia maintain their nuclei and their organelles, intracellular organelles, and are composed of the surface and intermediate layer instead. Here we can see the epithelial reedy ridges that goes down into the connective tissue. We see the connective tissue papillae that penetrates the epithelium. In red is the very thin layer called the basement lamina. The basement lamina helps the attachment of the oral epithelia to the lamina propria by intercellular bridges and junctions called hemidesmosomes, which lie on the basal lamina. Here we can see a section of keratinized epithelium, the uppermost layer, the granular layer, prickle cell layer, and the downmost basal layer. The second layer after the basal layer is the spinosum, which is called for their spiny appearance, as we can see due to cell shrinkage and the desmosomes here. So the cells have a star-like spiny appearance. Very characteristic spiny appearance of the cells in the spinosum layer. A section of keratinized epithelium with the full layers, keratinized, granular, and spinosum. We see the rete ridges and the connective tissue papillae. This part is the connective tissue papillae, and down is the reticular part of the connective tissue. This is a section of obviously a lining oral mucosa, oral epithelium, because we see a non-keratinized surface layer, superficial layer, intermediate layer, and spinous layer or prickle cell layer, followed by the basal cell layer. You can see very mildly or fewer rete ridges in here. As for the non-keratinized epithelium, we see more organelles in the surface layers, less tonofilaments, less developed tonofilaments, and there are no keratohaline granules present. This epithelium has greater permeability compared to the keratinized. Here is a section of para-keratinized or partially keratinized epithelium. The time taken for a progenitor or a stem cell to pass through the entire epithelial thickness and the upper layers and reaching the uppermost surface is called the turnover time. This is highest for the skin, and we have a very low turnover time of 5 to 6 days for the junctional epithelium. The non-keratinocytes are cells which compose only 10% of the epithelium. They are uniquely clear cells under the microscope, as you can see here. All cells that are clear can be non-keratinocytes. These cells can be Langerhans cells, Merkel cells, which are touch receptors, melanocytes, which synthesize the melanin pigment, or inflammatory cells such as lymphocytes. The melanocytes, as well as the Langerhans cells, are all dendritic cells which have long processes. Melanocytes and the Merkel cells, they are located mostly on the basal cell layer. The Merkel cells have sensory function, so they can attach to sensory neurons and fibers. Here is a section of keratinized epithelium composed of stratum corneum, stratum granulosum, stratum spinosum, and the stratum basale. The melanocytes are melanin synthesizing cells. They are dendritic and have long processes as marked by the black arrows. We see the melanosomes, which contain the melanin pigment also. These cells can contact up to 40 keratinocytes in what can be called an epidermal melanocyte unit, thus producing melanin and transporting it to keratinocytes, contributing to the color of the oral mucosa. A clinical picture of what can be called a racial pigmentation of the oral mucosa, mostly in dark-skinned individuals. It can be seen on the gingiva as well as on the buccal mucosa, the hard palate, and the tongue. In these individuals, we see a high activity of the melanocytes. The number of cells in both dark or fair-skinned individuals is the same, while what is different can be the melanocyte activity producing the melanosomes and their level of dispersion, their level of their size, and their degree of melanization. These individuals normally have high number of macrophages that ingest the melanosomes. Langerhans cells are antigen-presenting cells. They are also dendritic cells and have characteristic Berbick granules, which can be seen as marked by the arrows. These granules are rod-shaped and have immunologic function. These cells with their dendrocytes can ingest any bacteria and foreign antigens in the epithelium and can present them to the lymphocytes or the lymph nodes in the connective tissue. Therefore, they are migrating cells from the epithelium to the connective tissue or the lymph nodes nearby. Merkel cells are non-dendritic cells. They are often closely opposed to nerve fibers, as seen here, because they act as a receptor for sensory function. These cells are often seen deeply invaginated, and many small vesicles can be seen close to the nerve fiber. The lamina propria is also a part of the oral mucosa that is underneath the epithelium. For descriptive purposes, it is divided into papillary superficial layer and a reticular deep layer. The reticular layer contains bundles that are loosely attached of collagen fibers. Wholly, both layers are called lamina propria. The dense connective tissue underneath is the submucosa that contains blood cells, nerve fibers, and also minor salivary glands. The lamina propria, along with the submucosa, they constitute the mechanical protection and the nutrition for the epithelium. Epithelial connective tissue interface can be ultrastructurally divided into two layers, a lamina lucida and a lamina densa. This basement membrane has a characteristic hemidesmosomes that connect the basal cells with the connective tissue underneath. Along with the hemidesmosomal connection in the basement membrane, what keeps the attachment of the oral epithelium to the lamina propria is the undulated or the wavy surface of the interface. We see a scanning electron microscope the papilla of the connective tissue that are penetrating into the epithelial ridges. This interlocking between the epithelium and the connective tissue is what helps the ultrastructural attachment, along with the hemidesmosomal connections lying on the basal lamina. Moving to the subdivisions of the oral mucosa according to their function and their location. The oral mucosa is subdivided into three parts: the lining mucosa, which is most of the total surface of the mucosa, constituting 60%. This is the labial mucosa, buccal mucosa, alveolar mucosa, the floor of the mouth, the ventral surface of the tongue, and the soft palate. This mucosa functions little in the mastication, and therefore there are no high friction forces. It is therefore soft and pliable and non-keratinized. The masticatory mucosa, on the other side, constitutes 25% and composes the free and the attached gingiva along with the hard palate. Here it functions mostly with the mastication and has therefore to be rigid and rubbery texture to help in the food mastication. It is a keratinized epithelium. The specialized oral mucosa is 15% of the total surface. On the dorsal surface of the tongue, which is covered with cornified epithelial papillae, along with lingual tonsils, the vermilion border of the lip, and the junctional epithelium. The masticatory oral mucosa is the hard palate, regions of the hard palate, and the attached gingiva, marked with the black arrows. This gingiva is more whitish in color, referring to the keratinization that is taking place. This gingiva and hard palate are subjected to high forces of friction and mastication. A cross-section through the moving from the lip, the gingiva, the hard palate, and the soft palate. This site is the keratinized epithelium, and then we find the bone layer and another respiratory epithelium on the side of the nasal cavity. Masticatory mucosa of the hard palate. we see the epithelium, keratinized layer, pronounced reedy ridges, and underneath lamina propria. Another section of a keratinized epithelium of the hard palate, showing the keratinized layer. A distant section for a masticatory mucosa. This is the epithelium with the underlying thick lamina propria and underlying submucosa. Again, a keratinized epithelium and a thick lamina propria. The lining mucosa are areas subjected to high levels of distension and compression. It covers the lip, the labial mucosa, the buccal mucosa, and floor of the mouth, as well as the ventral surface of the tongue and soft palate. A section of the lining mucosa showing a partially keratinized epithelium, fewer reedy ridges that are less profound than the masticatory mucosa, and normally the lamina propria has more elastic fibers. We will go into more details in the next part of the lecture. Thank you for your attention.